Hi, I'm Suzi Ring, and I'm a healthcare reporter at Bloomberg News in London. Today, I have the great privilege of being joined by Professor Adrian Hill, the director of the Jenner Institute at the University of Oxford, one of the masterminds behind the COVID-19 vaccine. We're going to talk today about another vaccine that Professor Hill and his team are working on against cancer, and how in the wake of the pandemic, the discussion has really opened up about the potential for vaccines in other disease areas. So thank you so much, Professor Hill, for joining me today. Much appreciated. Do you want to just start by telling us a little bit about the vaccine you're working on and its genesis?
We have been working on vaccines specifically designed to trigger the cellular arm of the immune system for nearly 20 years now. And the COVID vaccine actually came out of that program. So we're using an adenoviral vector, as people know widely now, really to induce both antibodies and T cells. But we chose adenoviruses because, in our view, they were, and probably still are, the best way of getting very potent cellular immunity, particularly from CD8 T cells. And we were actually doing lots of studies in humans of cellular immunity and how we could induce it, not for cancer, not for COVID, but actually for malaria for many years. And there is an unusual situation where at one of the stages of the malaria parasite's life cycle, which is complex and I won't bore you with, only CD8 T cells will work. So that was a very good way of really focusing in on that arm of the immune system, which of course is the most potent way of designing a vaccine to tackle cancer, because these CD8 T cells are killer cells, and we worked out immunization regimes that would maximize their numbers circulating in the blood quite early on. And then that vaccine came to be used for many other things, including COVID. So where we are today is using that viral vector technology, as it's called, to induce a strong CD8 T cell response to cancer antigens, and trying to boost that to even higher levels by using a second viral vector. And you may remember that mix and match regimes like that are very much in vogue for for COVID immunization. What we've been doing over the last few years is focusing on prostate cancer. And we chose this because it's probably one of the more doable, achievable vaccines to make against cancer, because prostate cells grow pretty slowly. There are some good target antigens like PSA or STIAP or PAP and so on. And we've now put a cassette of those target antigens into those viral vectors. In one phase 2 trial that we've completed, we have looked at the immunogenicity and potential efficacy of a vaccine encoding a long-standing cancer antigen called 5T4 against patients with late-stage prostate cancer. And we've seen some early encouraging signals there in our university-run trial. But that program now has been handed over to the licensee of the chimp adenovirus technology, which is Vaccitech PLC, and they're undertaking 2 trials with the same technology, one of them in earlier stage prostate cancer, men who've, say, had surgery and are worried about recurrence, and vaccinating those as soon as there might be any hint of recurrence. And the other trial is an ambitious one with Cancer Research UK as well as Vaccitech, supporting it in late-stage lung cancer, where we'll take men with checkpoint inhibitors and also receiving, with their checkpoint inhibitor, chemotherapy, and adding a vaccine into that treatment regime.
You mentioned the decision to use the chimpanzee adenovirus because you saw it as the most potent vector. We're obviously seeing a lot of discussion now about the use of mRNA. You will obviously have the first marketed products using mRNA in light of COVID-19. What do you see as the benefits of using the adenovirus over mRNA? What are the pros and cons, and the decision behind taking your approach rather than an mRNA approach?
Yeah, well, a year ago, nobody could have answered that question because there weren't enough data published on RNA vaccines. We've now seen a lot, and they do induce, as we've seen, very potent antibody responses, particularly neutralizing antibodies. They do induce T cells as well, but it turns out that even though on an ELISpot assay you can see T cells induced early by an RNA vaccine, they're mainly CD4 T cells. There are some CD8 T cells, but certainly not as much as you would get with an an adenovirus. On the other hand, just in the last few months, there have been some studies where you give the adenovirus first and then give the RNA. And there it looks a bit more interesting because the adenovirus primes the CD8 T cell response. And it's always harder to prime than to boost an immune response with vaccines. And there are some worthwhile looking CD8 T cells. So maybe RNA will come into play as a booster regimen for cancer. And indeed, there's been one company on the West Coast, Gritstone, who have begun to assess that clinically. So the exciting thing about RNA is that it's new and we're still really learning how to use it. But for the moment, the most potent regimens remain a pair of viral vectors.
And you mentioned, obviously you and your team have been working on products like this for many years. In the public's mind, COVID-19 has really brought vaccines to the fore, and the idea of cancer vaccines, while we do know there are some prophylactic vaccines out there with HPV and others, seem quite a new innovation. Is this slightly hyped because of COVID-19? Can we expect to see results and a number of cancer vaccines, do you think, in the next few years? Or is it the reality that as per pre-COVID-19, these processes take a lot of time and we shouldn't get too excited about there being too much on the market in the near future?
So the way I'd answer that is to say what we've really learned from COVID-19 is that you can develop vaccines an awful lot faster than we routinely do. So 10 years is too long. That's not necessary. You should know whether your product is working or not in a shorter period of time. But what the really good news is that there is going to be more investment in vaccines, in vaccine companies, in vaccine technologies. And of course, cancer is one of the great prizes. And I'm sure we can vaccinate against cancer. As you mentioned a moment ago, we're able to do cancers where they're triggered by a viral antigen and prevent them. Probably some of the first cancer vaccines that will work really well will be cancers that have those viral antigens in them. So HPV, human papillomavirus, is looking promising. Hepatitis B as a cause of liver cancer is certainly of interest, particularly if you could get that early and clear the virus when the liver hasn't transformed to malignancy as yet. But, the ultimate goal is to be able to somehow break tolerance against the unique antigens that are in cancer cells. I think that's going to take a bit longer. But remember, the advantage of cancer vaccines is they don't have to work 80 or 90% like a COVID vaccine has been working. If you were to get 30%, 40% more efficacy by using viral vector vaccines, which, as we've seen in the last year, are not that difficult to produce and can be produced inexpensively, that would be a wonderful new approach in cancer that would not replace checkpoint inhibitors, would probably be used with them, would not replace other cancer therapies, but might be a very valuable new approach that would be very specific in its targeting. And remember, these vaccines we've been having are mild enough to give to healthy people, and they're fine a day or two later in all cases. And in cancer, this would be a very mild treatment.
And as you mentioned, we've learned from the pandemic that timelines can be accelerated, clinical trials can be run faster when needed. Is there any changes that you're taking in approach to the clinical trial that's going to be starting this year for the lung cancer patients for this vaccine?
No real changes. And in fact, that trial has been planned for a little while and is kicking off round about now. I think that what we are likely to see is a lot more interest. And of course, recruitment rates can be rate limiting in lots of cancer trials. I think now that the public are familiar with the power of vaccines, we'll have more people volunteering to participate in those trials, particularly if they have serious cancer. We're already seeing people with the smaller breast that's already showing a lot of interest, and I suspect those trials will go faster as a result.
And in terms of regimen, again, we're currently thinking about vaccines in the sense of you have maybe 2 doses. We're now looking at booster doses. When it comes to the context of cancer, what will we be talking? Is this something that would become part of someone's regular treatment forevermore? Is it something they would have temporarily every 6 months for a couple of years until a tumor had reduced? What would be the prognosis and the use of vaccines in that prognosis?
I think we'll be using vaccines differently. As you say, we're not going to be giving just, say, 2 shots and a month apart and then you're done. It's harder to vaccinate against cancer fundamentally because you're trying to deal with a self-antigen much of the time, or an antigen that's not very different from a self-antigen. So I think you will need rounds of vaccination. For example, with our 2 viral vectors, we give them as a pair some weeks apart, then come back maybe a few months later, give them again. And it may well be that we give multiple rounds and multiple immunizations and keep going to control cancer and to increase lifespan. I think that's a realistic early objective. We have the big advantage that we know what we're trying to do. We're trying to induce a very specific type of T cell that we can measure, and you can go and look for immunization regimens that maximize the number of T cells that are specific to the cancer. And one of the approaches that several companies are interested in now is not just giving an intramuscular immunization in a routine way, but actually giving the vaccine intravenously. Now, that would be very cumbersome for delivering a COVID vaccine, but of course it's entirely straightforward in a cancer patient who is already receiving intravenous checkpoint inhibitors and other medications. So that's certainly going to be explored. It's clear that there is a new way of immunizing that is not priming and boosting. It's called priming and targeting the T cells. So you give the first dose as a standard intramuscular immunization, and then you give the booster doses intravenously, and that actually brings the antigen that you want an immune response to the target tissue. So typically to the liver, but it could be to a mucosal surface, it could be to an accessible tumor, and then hopefully you would target in humans as well as in the experimental animals where this has been developed, those CD8 T cells to the tumor where they would kill it or at least slow its growth.
And as we discussed, there are some prophylactic vaccines out there already for HPV and others that you would see adolescents receive, for example. Is there anything in the therapeutic vaccine studies that could inform the development of prophylactic vaccines at all? Can we see links there? So if somebody had a predisposition to cancer for hereditary reasons or other, anything from the therapeutic side that can inform the prophylactic side?
Yes, it works both ways. The therapeutic side learns from the prophylactic side, and there are far more prophylactic than therapeutic vaccines. But we do more interesting things in the therapeutic space. I've mentioned one of them, changing the route of immunization, giving intravenous dosing. We also use more complex regimens, trying to really maximize the immune response that we can generate. And I'm pretty sure that the investment that we're seeing in cancer vaccines at the moment, which is substantial and growing, is going to have beneficial effects for vaccinating against viruses that we would like to prevent. So, for example, infectious mononucleosis is not the most serious infection we get in teenage years, except when it can turn into something that triggers cancer or multiple sclerosis. So it would be nice to have a vaccine against Epstein-Barr virus to stop us getting infected, as well as one that would target tumors like nasopharyngeal carcinoma, which is triggered by that viral infection and is a horrendously rapidly progressing tumor in many cases. So yes, lots of to and fro.
And we talked about cancer obviously today, but there seems to be kind of a growing list of diseases that could be treated using vaccination. We've all sort of become armchair vaccinologists during the pandemic. So I know there's a lot of focus on this right now. And for people like yourself, you've obviously been working on a number of these things for years. Do you see this as being a sort of key moment in time where we're going to just see the landscape grow incredibly in terms of the potential for vaccines?
I think that has started already. Some of the targets are extremely important medical conditions. For example, Alzheimer's disease, there have been vaccines tested in the clinic, not hugely successfully, but I think they will come. I think in 30 years' time, most research vaccinology will be in the space of treating chronic diseases and not treating microbes. I think we'll have covered most of the main microbes by then. And if you think about it, we're not doing with a vaccine very much that's different from giving a monoclonal antibody. In many areas, we are following monoclonal antibodies, which can be used to identify the exact target you want the immune response to go for. You have to keep giving those. There's not a durable immune response. So if you can identify the target antigens, say in a chronic disease condition, maybe a self-antigen with a monoclonal antibody that is shown to work, what a lot of vaccinologists are doing then is taking that antigen and developing it into a vaccine and generating a natural immune response rather than having to give a monoclonal antibody for perhaps many years.